So if you think about mammals, think about what's the most different mammal from a mouse. Often the answer I get is an elephant, and it's close. I'm going to tell you a story about mammoths. So mammoths were the subject of intense study, especially about 5 years ago, when there was a big interest in sequencing ancient DNA. And mammoths were a great target because they occurred in Siberia, and their DNA was essentially frozen in permafrost about 14,000 years ago. So this is almost like keeping DNA in a giant freezer, which is the best conditions possible. So about 5 years ago, the goal was to sequence an entire gene from an extinct organism. Now today, of course, we're sequencing entire genomes of extinct organisms, but just 5 years ago, we wanted to sequence an entire gene from the nuclear genome. So this is what a group in Germany, in Leipzig, headed up by Michael Hofreiter, did. And the gene they chose to study was the melanocortin 1 receptor, because it's a very simple gene. Remember, I told you we know a lot about its structure function. It's only about 1,000 base pairs in length, and there's no introns, so it's very simple, and it was a great starting point. So they sequenced the melanocortin 1 receptor in DNA extracted from mammoths. And here's what they found. They found a mutation. And what mutation was it? Well, it was an arginine to cysteine change at the exact same position that we found in beach mice. Now, of course, the DNA was extracted from bone, so they didn't know the phenotype or the coat color of mammoths. But based on our work in beach mice, what this suggests is that mammoths, like beach mice, may have been polymorphic in color. Now, the question of why they were polymorphic in color, we don't know the answer to. This could be due to survival differences. Others have suggested that it's due to sexual selection. So when this work was published, the press line was, "Blonde mammoths have more fun," but I'm not going to give you any explanation. I'll leave that up to your imagination. So in this case, we have very divergent organisms, mice and mammoths, that use not only the same gene, but the same mutation. And in fact, as I alluded to earlier, melanocortin 1 receptor is involved in a number of different color variants and a number of organisms that you may be familiar with. So in this case, it was the same mutation, but we know that other changes in the melanocortin receptor can also cause differences in color through different mutations. So I just wanted to give you some examples. So some work that I've been doing with a colleague at UC Berkeley, Erica Rosenblum, has shown that changes in the melanocortin 1 receptor are responsible for the production of these very adorable lizards that differ in color when they occur in White Sands, New Mexico. So you can guess which lizard occurs on White Sands and which one occurs off White Sands. Color differences are also involved in animals you may see every day, including cows. Also, many of you may know of or even have a Labrador dog. Well, they come in generally 2 colors. There's the black labs and blonde labs. Again, this is caused by a change in the melanocortin 1 receptor. And much more recently, again, work out of Germany, this time Svante Pabo's group, has shown that color changes in the melanocortin receptor, in addition to being responsible for human color differences, may also be responsible for color differences or hair differences in Neanderthals. So they showed that changes in the melanocortin 1 receptor or variants in the melanocortin 1 receptor were found in Neanderthals, suggesting that even back in those days there could have been redheads as well.